Thank you, Layla, for the introduction, and thank you, organizers, for inviting me to speak. I'm really excited to share with you all our recent work. So, mechanosensation is the process by which cells detect mechanical forces and then transduce them into downstream signals. The first example that many people think of is our sense of light touch, which is conveyed by specialized skin cells as well as DRG neurons, and is critical for interacting tactically with the world around us. But in addition to detecting forces extrinsic to our bodies, we also have the ability to detect many different types of internal forces that are also key to physiology. So, for example, chondrocytes in our bones sense the compression in our joints, endothelial cells in our blood vessels, and epithelial cells in our kidneys sense fluid flow, and even differentiation of neurons is regulated by the ability of these cells to detect their local environment, things like the stiffness or roughness of the local substrate. There's also many more examples, such as lung inflation, bladder stretch, and gut motility, just to name a few. And importantly, each of these processes that I've just listed that are initiated by mechanosensitive piezo ion channels, which are going to be the subject of my talk today. And the canonical view of how piezos play these roles in physiology is through ion flux or through ionotropic signaling. They're ion channels, so they open up and ions flow through their pore, and then those ions go on to do things in the cell. And for a couple of these processes, we do know that this is the case. So for example, in DRG neurons, piezos importantly are cation non-selective channels. So when they open, sodium and potassium ions flow in to depolarize or sodium and calcium ions flow in to depolarize the neuron and elicit action potentials. In a non-excitable cell, such as a red blood cell, it's actually the calcium influx. So when these cells undergo an osmotic stress, calcium enters the cell through piezo ion channels, activates the Gardos channel, which leads to potassium efflux and water efflux, and thus regulation of osmotic balance. But for a lot of other roles that piezo plays in physiology, we don't actually know that it's the ionotropic signaling that matters. And that's in large part because the field relies a lot on genetic knockout models, mouse knockout models. So you knock out the entire gene, the whole protein, and then see what the effect is on physiology. But when you knock out the gene, you're not just getting rid of ion flux, you're also getting rid of any other roles that protein might play. For example, its conformational changes, its physical interactions with other proteins. And indeed, some ion channels, including NMDA receptors and some subtypes of voltage-gated calcium channels, in addition to their well-established roles in ionotropic signaling, also can do what's called conformational signaling, where it's not just the ion permeation that is a relevant signal, but also the change in conformational state that is signaled to other proteins. And we hypothesized that piezos might be really good candidates for doing conformational signaling for the following reasons. So first, piezos are incredibly large transmembrane proteins. They're trimers, and each trimer has over 2,000 amino acids and 38 transmembrane domains. So they're really huge, and thus they take up a lot of membrane real estate, which really gives them a lot of opportunity to interact with other nearby membrane proteins. In addition to their large size, they actually undergo really large conformational changes. And so I'm briefly going to talk you through the gating cycle here. So on the left here, I'm showing a side-on view of piezo, and on the right, I'm showing a top-down view. And at rest, piezo sits in a bowl conformation, with the three blades kind of curling up and around a central cap domain. But in the presence of mechanical force, these blades flatten or sort of unfurl from this central cap domain, as you can appreciate in these beautiful morphs from Bailong Xiao's lab. This is accompanied by an expansion of the area of the blades, which you can appreciate from the video on the right. And so these large conformational changes again mean that piezos really might have a lot of opportunity to interact with other nearby proteins in the membrane. Now, additionally, the highly curved conformation of piezo on ion channels at rest, or in their closed state, actually has implications on the membrane far beyond where the membrane actually makes contact with the protein. And this is referred to in work by the Hasselwander and McKinnon labs as piezo's extended membrane footprint. So this idea that there's an energetic cost to this high curvature of the membrane, and that this curvature also extends far beyond the boundary of the channel itself. And we've predicted this footprint could actually reach up to about 100 nanometers away from the central pore of piezo. So this gives rise to the potential for piezo ion channels to interact with even more distal proteins in the membrane, not just those that it might directly bind to, but perhaps other proteins that might be sitting in regions of this highly curved membrane footprint. And finally, we already had one good idea for a potential target of piezo conformational signaling. And this comes from work a few years ago from Eric Honoré's lab. And I'll talk through these kinds of data more explicitly in just a second. But basically, what the Honoré lab found was that when you co-express piezo1 and the mechanosensitive K2P channel, TREC1, you get a pretty substantial potentiation of the TREC1 current. So you can see here on the y-axis is the current through TREC1, and it's much greater in the presence of piezo1 as a function of pressure in red than in its absence in green. And importantly, they found that this wasn't just true in an overexpression system, but also in a native system. So in a human adipose stem cell line that natively expresses both channels, if you measure the TREC current in a control cell, or you can see here in red, you get TREC currents that increase with pressure. But when you knock down piezo1 native expression with siRNA, this TREC current is nearly absent, suggesting that this piezo1 modulation of TREC1 exists in a physiological system. So we set out to specifically identify whether or not piezo1 was capable of conformational signaling using its modulation of TREC1 as a readout. So for the experiments I'm going to talk about today, I'm co-overexpressing the two channels in a mechanically silent cell line. So these are neuro2A cells in which their endogenous expression of piezo1 has been removed via CRISPR. And then to activate the channels, I make a cell-attached patch and apply negative pressure or suction through the patch pipette in a highly controlled and precise manner with a high-speed pressure clamp. Now, to isolate the activity of the channels from each other, I take advantage of their distinct reversal potentials. So I mentioned piezo1 is a cation non-selective channel, so it reverses at 0 millivolts. So in a cell expressing just piezo1 and a patch from a cell expressing piezo1, if I give a saturating pressure step, for example, to minus 80 millimeter mercury, at 0 millivolts we see no current, not because the channels aren't opening, but because we're at the reversal potential. Whereas at negative 80 millivolts, we see a large inward current that quickly inactivates, characteristic of piezo1's inactivation. Now, in a cell just expressing TREC1, we now see an outward potassium current at 0 millivolts because these are potassium selective channels, but nothing at their reversal potential at minus 80. And remarkably, when we look at a cell co-expressing both channels, we again see piezo currents, inward piezo currents at minus 80. And now, just like the Honoré lab, we observe that the TREC currents are much larger in the presence of piezo than in its absence, thus replicating this initial finding by the Honoré lab. And we quantify these data in several ways. First, we look at the peak current. And when we look across many, many patches, we see that on average, indeed, the peak currents through TREC1 are much larger in the presence of piezo. And I just want to highlight that this y-axis is on a log scale, so this is actually quite a substantial difference in peak current amplitudes. We can also appreciate this when we look at the TREC1 peak current as a function of pressure. And we can see that this persists across pressures, and in fact, the difference seems to grow with pressure. And finally, and the way I'm going to quantify this for much of the rest of the talk, we can look at this as the fold potentiation of TREC1 current by piezo1, or the ratio of the median currents in the presence and absence of piezo1. And what we find is that this plateaus around minus 60 millimeter mercury, right around where piezo currents plateau, at about a 5-fold potentiation of TREC1 current by piezo1. Now, you may have also noticed that the kinetics of the TREC1 current change. And so this we quantify as the steady state current, or the current remaining after 250 milliseconds of pressure. Of course, not quite all patches have completely saturated at this point, but I'll refer to this as the steady state current for simplicity. And what we find is that in the absence of piezo1, TREC1 currents desensitize quite rapidly, such that only about 5% of the current remains. But in the presence of piezo1, this desensitization is nearly absent in most patches, with a mean of about 80%. Okay, so now that we've replicated this initial result, we wanted to start to get at what the mechanism might be, and to specifically test if piezo1 might be doing this via conformational signaling. And as a first pass to sort of tease apart the difference between permeation and conformation, we took advantage of the fly isoform of piezo, which shares only about a 30% similarity with mouse piezo1. And importantly, fly piezo also is known from work by others to have a massively reduced single channel conductance compared to mouse. And so here I'm showing some data collected in my hands where we find that the fly single channel conductance is about 1/5 that is mouse. And appreciate the different scale bars here. So these single channel currents are actually quite tiny. We have to go to quite negative potentials to measure them. And importantly, both mouse and fly piezo we found via imaging experiments express at about the same levels, such that when we measure their macroscopic currents, the fly macroscopic currents are also about 5-fold lower than mouse. And so we reason that this would be a great first test of the importance of permeation. If permeating ions are mediating this effect, then we might expect that fly piezo would be less effective at potentiating TREC1 than mouse piezo. In contrast, what we actually found was, to our surprise, fly piezo was a much more effective modulator of TREC1. So here I'm showing example raw traces on the left. You can appreciate again that the fly piezo currents are smaller than that of mouse. And yet the TREC1 currents in the same patch are much, much larger. And this is also true across many patches, again highlighting that this y-axis is on a log scale such that this difference is fairly substantial. When we quantify this as the fold potentiation of TREC1, we find that whereas mouse potentiates TREC about 5-fold, fly piezo actually potentiates TREC1 upwards of 10-fold, again suggesting that it's a much more effective modulator, despite its reduced single channel and macroscopic currents. We also find it has a similar, if not slightly enhanced effect on the steady state current compared to mouse piezo. So in all, not only does fly piezo retain the ability to potentiate TREC1 despite the reduced current or the reduced number of ions permeating, it actually does a better job. And so this gives us our first hint that rather than permeation, conformational changes in piezo might be the more salient signal sensed by TREC1. To more carefully test for a role of permeation, we designed 2 additional experiments. So first, I want to highlight that when we're measuring TREC currents, we're measuring at the reversal potential of piezo. So there actually is no net ion flux through the channel. And yet, if you have calcium in your buffer, there still probably is some net calcium influx at this potential. So we repeated this experiment in calcium-free buffers. And importantly, in light pink here in the example trace and in the fold potentiation on the right, we find that there's no difference in the magnitude of TREC1 potentiation by piezo. So it's not calcium playing a role. To further test for a role of permeation, we took advantage of a lysine mutant, this piezo1-9K, that has 9 lysine residues introduced in the lateral exits or the lateral portals of the channel, and effectively turns piezo1 into a chloride selective channel instead of a cation non-selective channel. And again, we find in purple that there's no effect on the magnitude of permeation, or the magnitude of modulation. And so to summarize, the number and/or species of ions permeating really seem to have no influence on the modulatory effect, which suggests to us that it's not permeation. So then our next question was, well, if piezo1 doesn't have to permeate to modulate TREC, does it have to gate at all? That is, are conformational changes in piezo required for its effect on TREC1? And to answer this question, we took advantage of 2 double cysteine mutants that we had already designed in the lab that can be used to reversibly control gating via cysteine cross-linking. So the first pair links the cap of the protein to the blades of the protein. So we kind of think that it must prevent the blades from flattening. It locks the channel shut in that way. And then the second links 2 residues within the cap that are known to from simulations to move forward and apart during the gating cycle. And so in the absence of DTT, these residues are close enough to form disulfide bonds, and piezo does not respond to mechanical stimulus, so the channel is effectively locked shut. However, in the presence of DTT, the disulfide bond is reduced, and a mechanical stimulus again elicits a normal current. And importantly, this effect is both nearly instantaneous. It happens within the time resolution of our permeation system, which is a couple seconds, and it is also reversible. So we can wash DTT on and off and both lock and restore full gating. So I used these 2 mutants to test their ability to potentiate TREC1 both in the presence and absence of DTT. I'm only showing the raw data here for the cap to blade mutation, But what we found was that, again, in the absence of DTT, when the disulfide bond should be present, a stimulus does not evoke piezo currents. So again, the channel is conformationally restricted and it cannot gate. However, when I include DTT in my patch pipette, I again see a robust inward piezo current, and importantly, the TREC1 currents are again potentiated. And this is true both for the cap to blade mutation, as well as for the within cap mutation, this AP to CC. So in the presence of DTT, they both potentiate TREC equivalently to wild type piezo1. So what this experiment tells us is that full conformational flexibility of piezo1 is absolutely required for it to potentiate TREC1. So when the DTT is not present, it cannot potentiate. So even though the channel does not need to permeate, it does need to gate. It also tells us that the effect of piezo1 on TREC1 is relatively acute because we only apply DTT in our patch pipette about 30 seconds prior to applying the first mechanical stimulus. And therefore, it's not like there's some chronic effect of piezo1 being present in the cell that is influencing TREC1 activity or upregulating membrane expression, for example. All right, so in summary, the data I've shown you so far suggests that piezo fulfills all the hallmarks of conformational signaling. Conformational changes alone, and not permeation, can influence the activity of another protein. And so with these data in hand, we next wanted to start to try to get at the mechanism. And to begin to do this, we turned to an imaging approach. So all of the experiments I'm about to tell you about were performed by a very talented graduate student in the lab, Marie Cronin, who did super resolution STED microscopy on cells co-expressing piezo-MYC in cyan and TREC-HA in magenta. And so what I'm showing you here is a fly-through of a STED super resolution image of a neuro2A cell expressing both channels. And so the first question that we sought to answer with these experiments was, do TREC1 channels directly bind to piezo1? And the reason that we hypothesized that this might be a potential mechanism is that for both NMDA receptors and for voltage-gated calcium channels, the other ion channels that I brought up at the beginning that are known to undergo conformational signaling, in both cases, direct binding is the mechanism. And so for these experiments, we reasoned that based on the sizes of TREC1 and piezo1, as well as the locations of their introduced fluorophores, that the maximal distance of two puncta, or channels, if the two proteins were bound, would be about 30 nanometers, when also accounting for some linkage error and flexibility. And Marie also reasoned that if there is a binding affinity of piezo1 for TREC1, we would best be able to resolve this if we performed these experiments at low levels of piezo1 expression, in this case endogenous levels of piezo1 expression, and high levels of TREC1, so basically flooding the cell with TREC1. And so what Marie did is she engineered a neuro2A cell, which natively expresses piezo1, she CRISPR engineered it to have a MYC tag in piezo1 in the cap domain. And so what Marie did then was deconvolve and segment raw STED images to yield distributions of puncta that look like this, and then measured for each piezo1 channel in cyan, where is its nearest TREC1 neighbor in magenta? Again, with the idea of binding affinity would be seen within about 30 nanometers. So what these data look like for a single cell is you can plot a cumulative frequency distribution and then look at the nearest neighbor distances of piezo1 to TREC1. And so what we see is for this particular cell, on average, the two channels are actually quite far apart. For each piezo1 puncta, its nearest TREC1 punctum neighbor is probably about 300 nanometers away. And she summarizes these data across images. We can also bin them and then carefully inspect the first couple bins to see how many puncta have a TREC1 within about 30 nanometers. And so if you add up the proportion of piezo1 puncta across, I believe, 3 images, we find that only about 1% of piezo1 puncta have a TREC1 punctum within 30 nanometers, which suggests to us that there's no real hint of any stable binding interaction between piezo1 and TREC1, even when TREC1 is far in excess of piezo1. Okay, so now with the idea that they're not binding, we next had the question, well, what density of piezo1 channels is even needed for modulation? So we returned to our EPHIS data and we analyzed it in a different way. And so what we can do is for every single patch, we can use the surface area of the patch calibrated to the pipette resistance from experiments I've done previously, as well as count the number of channels in the patch based on the peak of the macroscopic current and the known single channel current of piezo 1, and actually count the channels per micron squared of piezo in each individual patch. And we can do the same for TREC 1. And then what we can do is we can plot the TREC 1 steady state current, which again is one of the measures of modulation. So these steady state currents are much larger in the presence of piezo than in its absence, and plot this as a function of piezo 1 density. And what we find is the following. We find that at low densities of piezo 1, we actually don't see much modulation at all. These data look remarkably similar to TREC 1 alone, with rapidly decaying currents. We see about a half maximal effect at about 5 piezo channels per micron squared, and then a saturating effect beyond 10 channels per micron squared, such that we have this nice sigmoidal relationship. So then with these data in hand, Marie went back to the scope and we asked a different question. Specifically, at densities of piezo 1 that are predicted to potentiate TREC 1 or not, how close are the channels? And so for these experiments, Marie subdivided the images, in part because the mean piezo 1 density across an entire image was often fairly low, not quite to saturation levels, right? So the density kind of varies across the cell. And so for analysis, what she did was for each TREC 1 puncta, she drew a 1 micron area circle around the point and calculated two things. First, the local piezo 1 density, simply by counting the number of puncta in the square, so for this, in the circle. So for this particular example, this would be a density of 5 channels per micron squared. And second, measuring the distance to the nearest neighbor, in this case, the punctum highlighted in yellow. And Marie then binned and plotted these data as a function of piezo density. And so as you might expect, if we plot the nearest neighbor for each TREC 1 to its nearest piezo 1 punctum as a function of piezo 1 density, the nearest neighbor decreases. And this is expected, right? So there's more piezos around at higher densities, and so it's easier to have a close nearest neighbor. But importantly, Marie also built a model to simulate random distributions of TREC 1 puncta in the same arena as piezo 1 puncta. And what she found was that there's a slight but systematic bias, whereby the empirical data show that TREC 1 is slightly closer on average to its nearest piezo 1 neighbor than you might expect. Okay, so now we wanted to look at these data in one final way and with one particular idea in hand. So at the beginning of the talk, I mentioned this idea that piezo creates what's called an extended membrane footprint, that its high curvature results in the membrane being curved far beyond the boundary of the membrane and protein interaction itself. And I also mentioned that we think that an upper bounds on the estimate for the size of this footprint would be about 100 nanometers. And so Marie specifically then asked at 3 different densities of piezo 1, can TREC 1 actually feel the piezo 1 footprint? Is TREC 1 predicted to be close enough to a piezo 1 to be located within an extended membrane footprint? So first she specifically looked at a piezo density of 1 puncta per micron squared. So TREC 1 puncta that had only 1 piezo within a micron. And remember, this is a density of piezos that is not predicted to potentiate TREC 1 because there's essentially no difference in steady state current between that and control. And so here, what we're plotting is the proportion of TREC 1 puncta that have a nearest neighbor distance within different bends of TREC 1 to piezo 1, with empirical data in black and simulated data in red. And what Marie found is that in this case, actually the empirical and simulated data look quite similar. And importantly, if you inspect just the first 2 bins here, only about 5% of TREC 1 channels are predicted to have a piezo 1 puncta within about 100 nanometers. So at densities that are not sufficient to potentiate, the vast majority of TREC 1 channels are not predicted to sit within a piezo 1 footprint. Next, if Marie looks at 5 puncta per micron squared, which is about a half maximal density required, we see 2 things. First, we start to see this left shift of the empirical data compared to the simulated data. So TREC 1 is slightly closer to piezo 1 than you might predict by random chance. And second, now if we add the first two bends to see how many TREC puncta are within 100 nanometers of piezo, we find that it's about 30%. So a fairly substantial proportion are starting to maybe sit within that piezo 1 footprint. And finally, if we look at densities that are saturating of 10 channels per micron squared that have the maximal effect on TREC1 steady state current, we again see this leftward shift, this bias in the empirical data compared to the simulated data. And remarkably, now we see if we add the first two bends that more than 50% of TREC1 channels have a piezo1 punctum within 100 nanometers. And so what this suggests to us is that the membrane footprint is certainly in play as a mechanism because the majority of TREC1 channels are predicted to sit within a piezo1 footprint. And so we think that this is one way that piezo1 might be communicating with TREC1. This is also a logical way that it might do so because TREC1 and other K2P and other mechanosensitive K2Ps are known to be sensitive to local membrane curvature. I do want to highlight, however, that by no means do we think that this is the only mechanism. So there are also other possibilities, including things like cytoskeletal network or local lipid modulation, that we're looking into in follow-up experiments to tease apart the role. All right, so to wrap up the data that I've talked about, first I showed you that piezo1 potentiates mechanosensitive TREC1 current amplitudes as well as eliminates their desensitization, basically replicating the result originally found by Eric on Array's lab. We found that this is true not only for mouse piezo1, but also for the fly piezo isoform, and that actually the fly isoform is more effective in doing so, which was a surprise to us. We also found that piezo1's modulation of TREC1 requires full conformational flexibility of piezo, but importantly, it doesn't require permeation. Neither the number nor the species of permeating ions seem to have any real effect on TREC1 modulation, suggesting to us that piezo1 fulfills all the hallmarks of an ion channel that can do conformational signaling. We also found that we don't see any evidence for piezo1 channels directly binding to TREC1 channels, even when TREC1 is in excess. And so this really sets piezo apart from the other ion channels that are known to do conformational signaling, in both cases of which binding is the mechanism by which they do so. And finally, we found that at high piezo1 densities, at densities that are sufficient to maximally potentiate TREC1, most TREC1 channels are within the piezo1 channel footprint, suggesting this is a potential mechanism by which piezo does this. And so I just want to take my last couple minutes to speculate a little bit about the role of conformational signaling in physiology. So I mentioned already that this has been found, this direct link between piezo1 and TREC1 has been found in at least one native cell line. In the same study, they also found that piezo1 modulates TREC in mouse gingival fibroblasts, and further, that both piezo and TREC knockouts have the same effect in a wound healing assay in this tissue. And so here, if you compare a piezo1 knockout mouse in green to a complete mechanosensitive K2P knockout in black, both have the same effect on wound healing, suggesting in physiology that these channels actually might work in concert on the same axis in this assay. Further, the Lowell lab showed recently that a non-conducting piezo1 mutant actually rescues a dendritic mistargeting phenotype in fly. So here on the left, you can see currents elicited from a wild-type piezo channel, and then the lack of currents elicited from this 2306 mix. So this is a non-conducting piezo mutant. It doesn't respond to mechanical force by eliciting a current. However, if you compare the percent of clones mistargeted and a piezo knockout here, you can see that this is rescued either by overexpression of wild-type piezo or, remarkably, by this non-conducting piezo mutant, suggesting that maybe in this system, it's actually conformational signaling that's sort of enabling piezo's role in dendritic targeting. I also want to note that while I've been talking today about piezo1 and TREC1, we know from experiments done by others that piezo2 is also capable of modulating TREC1, and that the other mechanosensitive K2Ps, specifically TREC2 and TRAC, are also targets of piezo1 conformational signaling. And so if you stop to think a little bit, that this gives you 6 combinatorial combinations of ion channels. And so if you imagine looking through the databases to see where potential combinations of these are co-expressed, you actually get quite a large variety of tissues, suggesting that the link between piezos and K2Ps could actually be present in a lot of different systems. And I also want to end by speculating that the mechanosensitive K2Ps are by no means the only target of potential piezo-1 conformational signaling. So again, I mentioned that we think that TREC1 and the mechanosensitive K2Ps might have been targets because of their known sensitivity to things like membrane tension and membrane curvature. There's a lot of other ion channels that are also sensitive to these things. So for example, in the mechanosensitive ion channel family, the TMM63 channels, which are closely related to the fly or to the plant OSFA homolog shown here, are vertebrate mechanosensitive ion channels that may also be targets of piezo-1 conformational signaling. And we also think that this could be broadly expanded to anything with a voltage sensor. So some voltage-gated ion channels are already known to be sensitive to modulation by mechanical force. And so when you expand this sort of potential list of targets to the entire voltage-gated family, it's really quite large. And so active experiments are underway in the lab to search for other targets of piezo-1 conformational signaling. And we just really think that this should expand the way that we think about the roles that piezo-1 plays in physiology. You know, I mentioned at the beginning that since we often use knockout models to sort of find evidence for a role of piezo-1 in physiology, we should expand our thinking to the role maybe not just being permeation, but also conformational changes, which is important for things like pharmacological targeting, suggesting, for example, that simply blocking the ion pore might not be enough. Okay, so with that, I would like to thank my mentor, Jorg Grandel. I would like to again highlight that Marie did all of the STED imaging that I showed, all of the acquisition and analysis. I would also like to thank the rest of the lab, as well as Duke and NIH for our sources of funding. And thank you again to the organizers for inviting me to speak about my work today. And with that, I would love to hear any questions.
All right, thank you so much, Amanda. That was a great talk. So for questions, of course, as always, you can write your questions in the chat, or if you would rather ask your question directly, you can raise your hand and Greg will make you a panel so that you can speak. Okay, so we'll start with the questions that are already in the chat that are quickly accumulating. All right, so there was a question asked very early on that got answered in the talk, so that was good. So then we have a question from Damien Bell, who says, lovely structure function studies, Dr. Lewis and Bob, thanks. In your A2328C to P23882C, the sulfide bridging studies, did you also see cadmium coordination effects and perhaps the various charged forms of cis-modulating MTA reagents would also give more information on steric, electrostatic, electrostatic and or salt bridging events at these substituted cis residues?
Yeah, so that's a great question. So the short answer is no. So we actually have not tested for any of those things, but I think it's a great idea. And in particular, I've specifically thought about MTS reagents, not for the alanine to proline linkage that you mentioned, but actually for the other one, the one that links the cap to the blades. We've thought about maybe like trying successively longer linkers between these to see sort of how far away from the cap the blades have to be able to sort of peel open for the channel to gate, but we have not done any of those experiments yet.
From Scott Zaweya, did the fly piezo have altered lower nearest neighbor distances to TREC?
Yeah, that's also a great question. And we have a new graduate student in the lab who is doing exactly those experiments, I think within the next couple of weeks. So hopefully we will have an answer soon, but it's a great question.
Then Zhangxi asks, thanks for the talk. Would you expect direct modulations of local membrane curvature, such as formation of flipodia or endocytotic pits, could activate TREC channels?
Yeah, so I would expect maybe. So I know, for example, that the cup former CPZ directly activates TREC channels. And so, which is part of what sort of got us thinking about this whole membrane footprint hypothesis. And so, um, I think my short answer is if not directly open, at least modulate. You know, the thing with TREC channels and other K2Ps is actually that they are so polymodal that a lot of things tend to open them. You know, I kind of think of their open probability as like teetering on a ledge, and a lot of different things can push it over the ledge. So I don't know if I would, you know, it's kind of hard to distinguish directly activate versus modulate the already existing open probability, but certainly I think so.
Question from David Cocoon. Do you think that other channels are sensitive to membrane curvature? The fact that nicotinic acetylcholine, glycine, and NMDA channels have consistent single-channel conductance from patch to patch suggests not, and even consistent kinetics. I'd expect that curvature might differ a lot from one patch to another.
Yeah, so that's an interesting question, one we talk about a lot. And specifically, what we talk a lot with piezo is the difference in scales of curvature. And so, you know, we also see that piezo, for example, has an identical single channel conductance patch to patch. The difference is that when you're talking about a cell attached or inside out patch, the scale of curvature is you know, maybe 10 to 50 fold bigger than the scale of curvature induced by a piezo channel, right? So the piezo channel has a radius of about 14 nanometers, and then the footprint, again, we think has a radius of up to about 100 nanometers, whereas when you're talking about an excised patch, you might be talking more about a radius of getting closer to a micron. And so I think just because a channel might behave the same in patches of different radii doesn't mean that it wouldn't be sensitive to the local curvature induced by piezo. It all just kind of becomes a question of scale.
Michael Young says, hi, Amanda, great work. At the spatial scale of a footprint, the local environment is probably quite heterogeneous, which may add variability to the distance between the two different channels, if it is in fact driven by some energetic minimum given piezo's extended footprint. Have you explored looking at a simplified system where the mechanical properties are likely more uniform to see if there is some optimal distance that TREC channels tend to settle in relative to piezos?
Yeah, so I think in that case, the ideal system really would be in a reconstituted bilayer or a reconstituted liposome. And we would love to do those experiments. And we just have not done them yet, in part because we don't routinely purify these proteins, but would love to collaborate with someone or get that up and running in the lab. Because I think really to tease apart the mechanism here, we need a more simplified system to answer these questions. That, for example, would directly answer this, for example, curvature versus cytoskeleton question. If you get rid of the cytoskeleton, you know, does this effect even still occur? And that will absolutely require a more simplified system.
Amanda, can I jump in there? I wondered if you've used MD approaches. So Mark Sampson and Stephen Tucker showed that you can activate TREC2 channels, at least in MD simulations, by changing the tension within the membrane. Could you incorporate both a TREC channel and a piezo channel into a bilayer in a MD simulation and see how the conformational changes in piezo affect the TREC channel?
Um, Yeah, I think that would be a really interesting way to look at the question. I actually don't know how tenable that is in an MD simulation. If at what point, because I know, you know, from talking with other people who do work with MD simulations on piezo routinely, you run into the problem that piezo is just such a large protein that it is computationally very, very expensive to do much with. And so I don't have a great answer to that, not being the expert in that, but I will say that I think has sort of limited some of those things.
Up to next question. Asser Beder says, great and interesting talk. Why do you think the fly isoform had more effect? Also, are there known human piezo1 mutations that are expected to affect this conformational signaling?
Yeah, so to answer the first question, we don't know. We have speculated. You know, there are no published structures of fly piezo, so I would be really curious to see how it differs from mouse piezo1 in terms of overall architecture. They are predicted to have the same number of transmembrane domains, and you know, piezo organizes in this sort of stereotypical bundle fashion, and they have the same number of bundles. Interestingly, we have looked at the AlphaFold structure of fly piezo, and it looks a little bit more curved than mouse. I know AlphaFold has all of the caveats that, you know, it has, but we've speculated that it might just be something about the different conformation. We are actively doing experiments in the lab. We have a new graduate student who is making a bunch of fly and mouse chimeras. So I think he's making 20 or so chimeras between the two channels to test to see if he can find where the difference comes from. And hopefully that will answer that question. As to your second question, are there known mutations that affect? Not that we know of, but we actually have not ever screened any. But actually, that's an interesting idea and maybe something we should look into.
Then Bertil Hill, this is very nice talk.
Thank you.
The next question from Bama Rankalawan. Great study. How would you dissociate the conformation and permeation functions of piezo-1? How would you test this?
Yeah, so that's a great question. And I think the easy answer is pharmacology. The difficulty with piezopharmacology is that it is not terribly specific. So the blocker that people use the most is ruthenium red, which unfortunately targets a lot of cation non-selective channels. And so that's not a great way to tease apart the two functions in vivo. Gadolinium also blocks piezo-1 and also blocks a lot of other channels. And so ideally, what we would do is actually create a mutant that we know is non-conducting and yet still does conformational signaling. So far, I have screened through about 12 different mutants that I tried to think might do so, mutations in the pore and elsewhere, and none of them allow us to dissociate that yet. But we're still actively working on that because I think that will be really important in physiology to sort of tease apart these roles. You know, our dream is to find a mutant that does not permeate, but still does conformational signaling, and then you can put that into a mouse and see which systems it affects. And I just have to keep screening to find that mutation first.
This question comes from Giovanni Jagesian. Interesting and great talk. I was curious if piezo-1 activation through its conformational change modulate other depolarizing cation channels, either by promoting excitability or by inversely suppressing them to protect the cells from overexcitation.
Yeah, so that's part of an ongoing screen that we have to test specifically voltage-gated channels to see if they're targets yet. And those experiments are underway. We haven't found one yet, but there's a lot of them out there to screen through. We've only tested a couple.
The question from Bertil Hilly, since you can label each channel with a dye, would you be able to use a Fred plate to see whether proximity increases with mechanical input?
Yeah, so we also would love to be able to do that experiment, and we have not tried. I think it would be a really interesting experiment. The reason we haven't tried yet is because of a fear, which may or may not be founded, that mechanical input will be hard to do in a microscopy setting, because to stimulate the membrane in that way, you necessarily have to perturb it, and things will move in and out of the plane. So for example, if you're stretching it, in a patch or something, you're introducing curvature, or the other assay that we often use to activate is poke. So we like literally poke the cell with a second pipette, and that is of course going to severely disrupt any imaging assay. So we just have to think of a way to mechanically stimulate the cell without, in a way that we can also do Fred imaging.
Next question from Barbara Niemeyer. Thanks for a great talk. Could the large conformational change of piezo lead to changes in lipid composition, potentially affecting activation of TREC?
Yeah, we certainly think that that might be one of the mechanisms. So piezo, in part because it's large, also is just predicted to have a lot of lipid binding sites. This is partly from MD studies. And we think this is a potential mechanism, and it's on our list of things to screen for.
Silong Yang says, wonderful talk. I was wondering, could repeated stretching the membrane reduce the fold of TREC1 currents, considering the inactivated form of piezo-1 may be flat?
Yeah, so that's an interesting question. We haven't looked at repeated stretching, other than of course when we produce the full P50 curves of this, we do stretch repeatedly at each pressure, but of course each time we wait 10 seconds in between pressure application to sort of let everything recover back to normal. So far in the experiments we've done, we can't sort of untangle whether it's the open versus inactivated forms of piezo-1 that do this, but it's an interesting question.
Next question from Guy Salama. Can you tell us more from an engineering point of view on the advantages of having piezo modulate track 1? Why does track 1 require such modulation?
Yeah, so I love this question. So I have two different sort of ways that I've thought about this too. Because when I first thought of this, of course, it's like, why do you have an excitatory channel and an inhibitory channel sort of working together? What is the point of that? And I think the answer depends on what kind of cell they're in. So I think in an excitable cell, it might be kind of similar to how voltage-gated sodium and potassium channels work together, right? So you have the initial burst of depolarization from piezo, but you don't want that to persist for too long. And so then you have TREC1 turning on as sort of a break to sort of restore you back to resting potential so you can get ready for your next action potential. In a non-excitable cell, my hypothesis is that actually TREC1 activity is predicted to hyperpolarize the cell. And in a non-excitable cell, the actual relevant signal is calcium. And so maybe that just increases the driving force on calcium just enough to sort of give you a little bit more signal. But that is all sort of purely speculation on my part, because I also kind of struggled with that at first. This idea that you've got two channels that work in opposite ways, why would you want them to sort of potentiate?
Next question from Eleanor Mitchell. Great talk. Do you think that the TREC1 potentiation could be lipid mediated? TREC1 is also activated by PIP2, for example.
Yeah, so again, I think that lipid modulation is certainly on the table, and that's something we're looking into. Again, the large size of piezo means that there's not like one obvious lipid binding site that we can just mutate and then repeat the experiments. But certainly PIP2 cholesterol is another molecule that we've thought about, and yeah, it's on our list of potential mechanisms.
David Colquhoun, thank you for the answer for the previous question. Another question from David Colquhoun that's not directly to do with ion channels, but I'm curious to know whether any of you have had any NIH grants withdrawn. Maybe we can write those answers in the chat if you'd like. We have comment from Philippe Gilly. Beautiful work and excellent talk. Thank you.
Thanks.
And a question from Sergei Polchevsky. Great presentation. Can we assume something about possible evolutionary ancestors of piezo channels?
Yeah, so, we've at least shown here that it extends as low as fly piezo. To my knowledge, I don't think there's anything lower than fly that has actually successfully been cloned and tested in a heterologous system. So I don't know that I really have much to speak to beyond that. But I think certainly we have evidence both from my experiments showing that fly potentiates TREC, as well as the experiments from the Wool lab showing, that in that fly dendritic mistargeting assay that a non-conducting piezo mutant rescued the phenotype, that this must persist at least as early as fly. But I don't really have much to say earlier than
that. Yes, the chat is disabled because we're in a webinar setting. So the only place to write comments is in the Q&A box, which everyone can see, right? Yeah.
Can I ask a question, Leila, while we work on it?
Of course, yes. Go ahead.
Sorry if I missed this, Amanda, but how reciprocal is this regulation? So if you can activate TREC by pressure or tension or membrane stretch, can you change the properties of piezo?
That's a great question. So the answer to that is mostly no, except that we did see a slight effect of TREC1 on piezo1 inactivation kinetics. So we see no change in macroscopic current amplitude. We see no change in P50, so the sort of the curve, the pressure curve by which piezo is activated. But we do see subtle and reproducible changes in inactivation kinetics. And we haven't yet followed up on that further, but it's sort of something that's lurked in the back of my mind is an interesting way in which this effect might actually be reciprocal. A lot of change piezo inactivation kinetics, though, so hard to say how specific that effect is.
In my PhD, when I was in Stephen Tucker's lab, we were trying to work out how TREC2 channels change conformation with membrane tension. And so we found some mutations which we think shift the conformational equilibrium between a down and an up state of the channel. So it might be interesting to take some of those mutations which promote this up state of the channel, which we proposed was correlated with the mechanoactivation of the channel, and see if they have a different effect on piezo channel gating than the
that would be
wild type
super
channel. Right?
interesting. Yep.
But I was wondering, and I'm sorry, you may have already said this in your talk, but I was wondering how the piezo inactivation figures in the activation of TREC.
Right. So we
Say
think
that
that so we don't have any experiments that we've done to directly test this, but based on the fact that TREC1 desensitization is absent, you know, the TREC1 currents are still large and robust and non-inactivating long after piezo1 has completely inactivated. So we speculate that the inactivated state must be at least as efficient as the open state at doing this modulation, which again sort of hints against the role of permeation, right? That it must be something about conformation. And I think the prevailing assumption is that the inactivated state, based on some recent structures from Bailong Xiao, is still relatively flat. It just has a slightly different cap location.
Yeah, I guess that's what I was really asking about.
Yeah. But we would love to, for example, find like a cross-linking pair that sort of traps the channel in an inactivated state or something, and then we could directly test for a role of that.
Amanda, thank you for a great talk, and that was
thank you again to the organizers for the invitation. This was a lot of fun.